Hey learners, welcome to Ahmad Coaching and I'm your teacher Dr. Anam. Today we will study about gene sequencing Sanger method. Gene sequencing is actually a general laboratory technique which is used to determine the exact sequence of nucleotides or bases in a DNA molecule. We know that there are four different nucleotides: A, T, C and G. Now, through gene sequencing, we find out what is the sequence or how these four nucleotides they are arranged. Why we need to determine the sequence of the gene? Because the sequence of bases it actually encodes the biological information that cells use to develop as well as to operate. So all the information, how your cell is going to operate, how you are going to look, how you're going to perform your function is encoded in the genes. So if you determine the sequence of nucleotides in a particular gene, it means you are actually studying that gene. Now, how you can do the gene sequencing? The main principle is to generate pieces of DNA of different sizes, all starting from the same point but ending at different points. For example, I have a DNA and I want to do the sequencing. What I am going to do is that I am going to start from the same point and then I am going to generate different size pieces which are going to end at different points like this. So here you can see that I have 1, 2 and 3 different pieces of DNA, all starting from the same point but ending at the different point. After that, these sequences they are separated on the agarose gel. Agarose gel is actually a special gel through which the DNA molecules they pass and then they are going to be separated based on their size. After the separation, I'm going to read the sequence from the gel and then I can determine, okay, here we have A, here we have T, here we have C, and in this way I can find out the sequence of gene. There are different gene sequencing methods, but we are going to study about Sanger method here. The Sanger method was proposed by Sanger in 1977. It is also known as chain termination method. So if you say Sanger method or if you say chain termination method, both are same. Why this method is called chain termination method? Because in this method dideoxyribonucleotides they are used to terminate DNA synthesis at different sites. I told you that we have to generate different size species. Similarly, what here happening is that when the DNA is synthesizing, we are going to stop its synthesis at different size like this, and because we are terminating the chain like this, okay, from here no further movement is required. So as we are terminating the chain synthesis, that is why we are calling this method as chain termination method. Talking about the dideoxyribonucleotide, normally in DNA we have deoxynucleotide, in which here we have OH molecule, and this OH molecule is going to help in the binding of upcoming nucleotide. It means that next nucleotide is going to bind here. But when you are going to use dideoxyribonucleotide, at this position we have H, and because of the presence of H, no new nucleotide can be added at this position. So whenever, for example, here we were having A, T, C, and then we have this D, D and TP. It means that from here to onward, no more nucleotide is going to be added and our chain is going to be terminated. Here, this is the diagram or flow of Sanger method. In the first step, we are going to have a single stranded DNA with unknown sequence. Why the sequence is unknown? Because we are going to find out its sequence. You can see this is the sequence. Then we have DNA polymerase, and then we have the 4 nucleotides. We are actually performing PCR reaction here. In the PCR, we are going to make the copies of this single nucleotide, and when the PCR is going to make the copies, we are going to insert our this D, D NTPs or our dideoxyribonucleotides. So whenever the dideoxyribonucleotide is going to add, the termination of the synthesis, it will takes place. So in this way we have prepared 4 different reactions, and in each reaction we have added 4 different nucleotides. Here we have adenine, here we have cytosine and guanine, and all these have DD. It means that they are dideoxy, or they have H in this position. So when the reaction it will run, wherever these DD NTP will come, we can see that the reaction is stopped. Here, here and here we can see that we have DDA, DDA and DDA. So it means that we are actually generating different size fragments. Now we have longer size fragments, we have shorter size fragments. After that we are going to load these samples on the agarose gel. This is our gel. This sample is loaded here, this sample here, this sample here and this sample here. Now I told you that the samples or the fragments, they are going to move according to their size. So they are going to move from the longer to the shorter size. It means that small fragments will be here, while the longer fragments, they will be here. So we can see that longer fragments, they are here, 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 and in this way they are moving. Now what we are going to do, we are going to actually start reading our sequence. The first band here is representing G, so we have G. After that we can see that there is no band in this lane, there is no band in this lane, and then we have this band which is of a nucleotide, so we read A. It is like a stair, you are reading the stairs, okay? So the next band is here in the C lane, so we have C. The next band is at T, so we have T. Then again next band is at G, we got G. Here we have A, we got A. Again the next band is at A, means we have A. Then we have G, and then we have last band is at C. So it means that this is our row sequence which we studied from the longer reads to the smaller reads. It means that you are going to read your gel from top to the bottom, and then after that you can find out its anti-sequence, or you can find out the second sequence or the second strand, because we know that G is always going to bind with C, A with T, C with G, T with A. So in this way you can find out the double-stranded structure of unknown sequence by the help of Sanger method or chain termination method. So that was all about gene sequencing Sanger method. If you have any question related to this topic, you can ask me in the comment section. I will see you in the next video.